In this video, we're going to look at hearing. And so for hearing, we need to look at the ear. The ear can be divided into three distinct regions called the external ear, the middle ear, and then the inner ear. And each of these parts of the ear is either associated with hearing or balance or both. And I'm going to include a different video for balance, so I'm not going to talk about it a lot here. I just want to kind of make you aware of where in the ear we do have balance, because that is an important sensation that our ears are also responsible for. So we'll walk through these different regions, and I'll point out when we get to balance. So first, for the external ear, this is from the auricule, which is the part of the ear that you're familiar with. It kind of forms the shape of a funnel to help to funnel the sound waves through the auditory canal. This is called the external auditory canal, and it passes through this hole in the skull called the external auditory meatus, meatus meaning a hole, so it's a hole in the temporal bone of the skull. So the auditory canal passes through. This is all the areas that you could stick a Q-tip, although you're really not supposed to stick a Q-tip because it'll push all that earwax and impact your eardrum and make it so that the eardrum cannot vibrate. But this whole ear canal will get coated with wax, which actually helps to protect and kind of trap any foreign bodies that try to make their way towards your eardrum. The end of the external ear is at the eardrum. So we're seeing the eardrum in the picture right here. The eardrum is going to also be called the tympanic membrane. And this is a flat, thin sheet of tissue that vibrates with sound waves, kind of like the top of a drum. Okay, and then after the eardrum, we get to this other small little region called the middle ear. The middle ear is and should be an air-filled chamber. If it gets filled with fluid, the fluid will drain out via the auditory tube, which is connected to your throat. So that would be able to drain off of there. Also inside the middle ear are going to be the three ossicles, which are the three bones of hearing. So the three ossicles are called the malleus, the incus, and the stapes. In that order, spells miss. And the idea of these three bones is the malleus is attached to the eardrum. And so when the eardrum vibrates, the malleus starts swinging. So when the eardrum vibrates, this malleus is swinging back and forth. And the malleus is connected to the incus, which is connected to the stapes. So what these three ear bones do is they amplify the vibrations that are hitting the tympanic membrane. They kind of cause a bigger swing and cause more impact then. And what they're doing is they're conducting these sound waves from the eardrum to what's called the oval window. So the oval window is going to be part of the inner ear. And the oval window a little bit is kind of like the tympanic membrane, but it's actually sealing the cochlea, which is what we're going to look at next. So these malleus, incus, and stapes are part of a machine, basically, that's going to amplify the vibrations that are hitting the eardrum so that the vibrations are even more extreme within the cochlea. So that's the middle ear. The inner ear is going to include a couple regions. It's going to include what's called the cochlea, and then the vestibule, and then the semicircular canals. The inner ear is the one region that is for hearing and balance. And the reason that it also includes balance is because the vestibule and semicircular canals are specific for balance. And so I'm going to link in another little video that will talk a little bit more about the semicircular canals and their role in balance. But once we see how some of the features of the cochlea work for picking up vibrations for hearing, it's a little bit similar to what happens in the semicircular canals for balance. So the cochlea, which is this region here that kind of looks like a snail shell, this is where we get our hearing. So over here, this were the semicircular canals, and in the middle here, this region is the vestibule. But it's the cochlea that we're mostly going to focus on because the cochlea is specifically for hearing. Now, all of these structures in the inner ear are full of fluid, and the fluid is going to pick up these waves that are created by sound or are created as you disturb the balance for those in the semicircular canals. So because the inner ear is really where we're picking up hearing, we're going to focus straight into the inner ear. So now we've zoomed in, and we can see more clearly. This is the vestibule, this is the cochlea, and these are the semicircular canals. They're each oriented at a different angle. And this right here is the oval window. So the oval window is connected to, I'm going to go back. The oval window is connected to the malleus, the incus, and the stapes. So this region of the stapes actually vibrates against the oval window. And the idea of each of these bones, they're linked, each of these bones for the auditory ossicles, the auditory ossicle bones, are joined by synovial joints, which is kind of funny to think about. It's the same type of joint that you have in your knee or your elbow, these synovial joints, which allow a lot of movement between the bones. But so vibrations hitting the malleus will lead to vibrations hitting the incus, which then causes vibrations in the stapes, and the stapes presses directly against the oval window. The oval window then is linked to the vestibule and then the cochlea. So the cochlea is where we have our hearing. And the cochlea itself, really all of these inner ear structures, are nestled within the skull bone. So they're literally within the skull bone, and you can kind of see these features of the skull bone having a lot of these sponginess or kind of these holy bones that are able to also pick up vibrations. Now, this inner ear is going to have or be called the bony labyrinth. So this bony structure is what the cochlea sits in. And inside this bony structure is going to be the membranous labyrinth. So here in pink, we're seeing the membranous labyrinth. And the membranous labyrinth is really what we want to follow in order to understand hearing. We can also see that there's space between, this is the bony labyrinth, that all of this is setting inside, and this is the membranous labyrinth. So there's fluid in between these structures. And this fluid is called the perilymph. And so that's this fluid that's in these regions here, kind of shown in blue. The membranous labyrinth itself is also filled with fluid called the endolymph. And it'll become relevant later. The perilymph is kind of our typical fluid we see elsewhere, the cerebral spinal fluid. But the endolymph actually has higher potassium than sodium. And that's only going to be relevant because we're going to talk about potassium then entering cells and causing depolarization instead of sodium, which is a little bit weird, but it's because this fluid that's surrounding the cells we're going to be looking at has a really high potassium concentration. So that's why we're going to see that difference later. Okay, so let's try to further orient to what we're seeing in these inner ear structures. Here is that oval window again, and just beyond the oval window you start this long path, which includes, this is the bony labyrinth, and then this is that membranous labyrinth. And then these areas which we said were full of perilymph between the bony labyrinth and the membranous labyrinth each have their own names. So this here superior is called the scala vestibuli, and this below is called the scala tympani. So we name each of these regions. This is a T, this one's a V. But this in the middle, the membranous labyrinth, is also called the cochlear duct, or the cochlear duct is part of the membranous labyrinth. And for the scala vestibule, or the scala vestibuli, it has this bottom layer called the vestibular membrane. So that's this region. It forms kind of the top of the membranous labyrinth. And then the scala tympani includes this membrane, which would be the bottom of the membranous area. And this basilar membrane is going to be extremely important for hearing. So when this vibrates, this is going to actually vibrate our cochlear duct cells, which will then pick it up and have this effect of hearing. So Another thing to orient here is the scala vestibuli is technically from the oval window to the helicotrema, which is the apex of the cochlea. So if the cochlea goes like this here in the middle, this is the apex. And what I'm telling you is the scala vestibuli begins at the oval window and ends officially when you get to that apex. So that's the scala vestibuli. Then the scala tympani, which is the bottom part here. So this was the scala vestibuli. This is the scala tympani. I'm going to erase those so you can see them again. It begins, or we could maybe trace its path as beginning at the helicotrema at the apex, and then going back down, and it ends at what's called the round window. And so this is kind of the path of vibration as it enters the inner ear. It first causes vibrations and fluids within the scala vestibuli that goes all the way through until you get to the apex. And then those vibrations continue in the scala tympani, which goes all the way back through and pushes out of the round window. This round window has to also be flexible so that those sound waves can kind of push out instead of then reverberating back inside the ear. So these two areas, the scala vestibuli and the scala tympani, are parallel to each other and go all the way through the cochlea, but they kind of form these two discrete paths of sound waves, coming in to the apex and then going back out to the round window, coming in through the oval window and leaving through the round window. And the idea is as these sound waves are moving through, they're going to be causing vibrations in this middle area called the cochlear duct. And the cochlear duct is what we really want to follow as we continue. So remember, for the cochlear duct, it has this vestibular membrane that we no longer care about. And then it has the basilar membrane, which I actually care a lot about because this basilar membrane is going to be very important for sound. So let's follow the basilar membrane in the next picture. So here we've zoomed in directly on this cochlear duct with the scala vestibularly above and the scala tympani below, and this basilar membrane being the one that we're focused on. Now the basilar membrane is going to be these collagen fibers that are stretched from the spiral lamina, which is this bony extension here. So these collagen fibers attach here to the spiral lamina and then extend across and attach at the other side to another bony structure called the spiral ligament. And so these collagen fibers are going to be stretched across here, forming the basilar membrane. So these collagen fibers are kind of like the strings on a piano, because what happens as we move along the cochlea, here we're seeing the spiral formation of the cochlea. This is actually showing us the cochlear duct region, which is between the scala vestibuli and the scala tympani all along the cochlea. And here where it enters, the cochlear duct is very narrow or very thin. And then as you continue along the length, here at the middle near the apex, the basilar membrane or the cochlear duct is actually very wide. So the length of these collagen fibers, when you look at different areas of the cochlear duct, those ones near the oval window are going to be very short. Those ones near the apex are going to be very long. And this is kind of like the strings on the piano. The thick, short collagen fibers are going to vibrate with high frequency sound waves, and those thin, long fibers are going to respond to slower wavelengths of sound. And everywhere in between, they actually tune to specific wavelengths that they vibrate with. So what we're saying is that these collagen fibers that stretch across from the spiral ligament bone, or what's going to be the bone, to the spiral lamina bone, these collagen fibers are different lengths. In some places they're short and thick, in other places they're long and skinny, and that's going to cause different vibrations depending on the sound frequencies that you are listening to. Sitting on top of this vibrating basilar membrane all along all the regions of it, the short regions and the longer regions, you're going to have these same types of cells. These are the specialized cells that form what is called the spiral organ, and these are sensory hair cells, which is a little bit like maybe what we saw for the taste hairs and those other types. These are going to end up being pretty different. But these are specialized hair cells. So here we're seeing the cell. Look, it's a typical cell. It's got a nucleus. What a hair cell has that causes it maybe to have the name is it has these cilia, or these membrane extensions at one end of it. These cilia are really specialized. They're called Stereocilium. And we're going to see the types of things that are in the stereocilia that cause them to depolarize in response to sound waves. So first, in this picture, this was our basilar membrane, which could be different lengths at different regions of the cochlea. And sitting on top of the basilar membrane, we have various epithelial type cells. And then nested among them are these hair cells. So here's a hair cell, and here's one, and then here's one. So there's these row of hair cells. And technically these outer rows, these 3 cells here that we see, these are supporting cells. They regulate the tension of the basilar membrane. So they kind of tune this region of the basilar membrane. And then this inner row, so this cell here, is the one that's actually responsible for hearing. But all of these are going to be connected to the sensory neurons that are going to be a part of that vestibular nerve. Okay, so the tips of the stereocilia, so these little stereocilia that are at the ends of the hair cells, their tips is embedded in what's called the tectorial membrane. So this here is the tectorial membrane, which is acellular. It's kind of a gelatinous matrix. And what it's really going to do is it's going to be firm and non-moving. And as the basilar membrane vibrates, these hair cells are going to be bumping into this non-moving tectorial membrane. And when the hair cells, because of the vibrating basilar membrane, when they bump into this is going to cause the stereocilia to bend. And in the next picture, we're going to look at what happens when these stereocilia bend, because the whole basilar membrane is vibrating and pushing them into this gelatinous mass, which is called the tectorial membrane. So let's zoom in. So now we're looking at the stereocilia on the hair cells. Just to kind of orient you, I'm going to draw that hair cell. Here's its stereocilia kind of sticking up from it. And what you'll note in this picture, which is a lot better than mine, one stereocilia will be next to another stereocilia, and there's always a longer one behind it. So they're kind of very carefully shaped. And then between the two stereocilia is what is called a tip link. And these are really cool. So this is a stereocilia. And then this is a longer stereocilia next to it. And then this is the tip link in between. And let's zoom in on it. So in one membrane, in this membrane, we have the gated ion channel. And here is the gate. And then in the other stereocilia, right here, we have a protein that's connected to another protein that's connected to the gate. And what happens is when that basilar membrane vibrates, this hair cell, or remember that tectorial membrane is right here. And so when these guys vibrate, this guy gets pushed against it and maybe deflected backward, and it causes tension between these two stereocilia, which causes this stereocilia to pull on the spring that's attached to the gate. And so as these stereocilia bend next to each other, they literally pull open that gate, allowing this ion channel to open. And remember I said it's not going to be sodium that rushes in, because this fluid actually has higher potassium than sodium. So technically potassium rushes in, and that depolarizes the cells, which this stereocilia is part of. This is the hair cell, which is important for hearing. And so as potassium rushes in here, it can depolarize the whole cell. So this is called a gating spring that opens the potassium ion channel and causes the hair cell to be depolarized. Okay, so to kind of sum up what we've said that sound waves that are in the scala tympani, so down here, will push on the basilar membrane, and as you have sound waves hitting the basilar membrane, it pushes these hair cells up against this tectorial membrane that does not move. So as these guys are pushed up against it, it bends these little stereocilia, which are part of the hair cells, causing the hair cells to depolarize as those little tip links open and potassium rushes in, depolarizing the hair cells. So as we bend them with the basilar membrane, these guys depolarize, and they are going to release neurotransmitters to these yellow axons, which are synapsing with these hair cells. Because the hair cells themselves do not have axons, but these sensory neurons directly behind them do. So these are the sensory neurons. The depolarized hair cells release a neurotransmitter, which is an excitatory neurotransmitter called glutamate, which will then excite the sensory axon terminal. So here, these are the sensory axon terminals. They're going to be receiving glutamate, which is a neurotransmitter, and which will cause them to then have an action potential and carry that stimulation up this nerve, which is called the cochlear nerve, as it leaves the cochlea, but then it's going to join with the vestibular nerve, which is coming from the semicircular canals and vestibule for balance, and those together will form the vestibulocochlear nerve. And that vestibular cochlear nerve is the 8th cranial nerve, and it'll then enter via a region of the brainstem and eventually map to an area of the cortex called the auditory cortex, which tends to be, I think of it as being kind of the temporal lobe, though you can see in the picture, technically this is the temporal lobe, and so all of the sound is not directly on the temporal lobe, but it's all kind of near. We have these different regions of the cortex which are going to interpret sound waves. We have including the Broca and Wernicke's area, which are associated with speech. But so this is where those neurons will then map their auditory sensations to. And it looks like I've done all this recording and I've not come up with a password, so let me give you a password now. Our password for this week's video and mini test will be the word cochlea. So cochlea is the password, C-O-C-H-L-E-A, cochlea.